Hey everyone, Mr. Murphy here. I'm going to continue with section 4.4, which is all about the cytoskeleton. So the cytoskeleton is a component of the cell which we are probably a little bit less familiar with. Perhaps on a broad definition, you're aware that it provides structure. It helps with organelle movement and positioning inside of the cell. Cytoplasmic streaming, which is a cool concept. And then really just all of the support and structure that the cell needs relies upon different cytoskeleton components. So there are three main types of cytoskeleton components, which are broken down into microfilaments, intermediate filaments, and microtubules. Now, throughout this, there's not a lot of specific knowledge which we need to retain, like in terms of names of things, but I want you to be familiar with the general role that these different types of molecules play within the cells, so that we can work with information about these things using just the more general cell topics that we've gone over in this chapter. So just first off, so that you are aware, these are in order from sort of smallest to largest. In terms of structures, they're all on a nanometer scale, but microfilaments are significantly smaller than intermediate filaments, which are less developed even than microtubules. So some of these will come back to be a little bit more important when we go over things like mitosis and meiosis, as the cytoskeleton, being spindle fibers specifically, are a component of the cytoskeleton, which is massively involved in the process of sorting those chromosomes and things. So microfilaments, these help a cell itself, or maybe parts of a cell, to move. Depends on if we're looking at like single-celled organisms, which of course have a little bit different needs than the cells throughout our own body. They help to determine the cell's shape. Cells, are these sort of blobs that without specific structural components, they're simply going to collapse under their own weight or be easily influenced by the things around them. And if you think about inside of our body, we have trillions of cells associated with different organ systems and things, and we need those cells to hold a particular shape in order to serve their function appropriately. Microfilaments are made from a protein called actin. Actin, again, there's not a lot of specific terms which we need to be aware of. Actin is a somewhat important one in biology because you're going to see it pop up all over the place. So being familiar enough to recognize what that's referring to is a helpful thing. And then these can be made shorter or longer, and the process of that is kind of cool. They don't they can actually extend from both sides by being built up or broken down from the ends. And so they're not really like stretching or growing in a traditional sense like we might think, but rather they're being actively built up and actively broken down. And this is a process called dynamic instability. And basically the actin polymer is made up of different little tiny actin subunits that get added on to either end. It's a much more complicated process as you dig into it. That's not super important for us specifically in this course, but understanding that this term of dynamic instability, I think, is a valuable thing just to help us to conceptualize what's going on. In muscle cells, the actin and myosin are two proteins which actively work together, and basically what's happening there is that when your muscle cells contract, there's these little units where there's myosin heads that are pulling on this actin, and it pulls these two actins basically into the middle. And so there's fixed ends to these things. So as the actin gets pulled by the myosin, you see how it contracts, it condenses, and you get thousands of these units all in a line together throughout muscle cells, and that can add up to the contraction and elongation of major body muscles, which is good stuff. So here you can see actin. It's 7 nanometers wide, tiny thing, but within a cell here, there's a nucleus, and we see microfilaments stained in this picture here, and they are all over the place throughout this cell. So they serve a whole lot of purposes. Intermediate filaments, a little bit bigger. You don't need to write any of this down and know specifics, but just to help you understand, again, some major concepts within this class, there's at least 50 different kinds in any given cell. And so just to give us a sense of like how much stuff is actually going on inside of the cell, how much chemistry is going on inside of the cell, it's mind-blowing as we continue to understand more about how cells perform every single little nuanced function of what the cell needs to do to survive. We realize that there are many, many components that make up the ability to do that. So these are a little bit tougher. They're rope-like protein appendages, more permanent, and so they don't show that dynamic instability. They don't grow and shrink in the same way that actin and microfilaments do. These are a little bit more about anchoring and maintaining sort of a consistent thing. So we'll see in our next chapter, or actually at the end of this chapter, we'll see some intermediate filaments involved with how cells can adhere to one another and form these larger tissues and organ structures. And so you can see intermediate filaments, they're not so much about pulling and pushing things around, but rather about just maintaining a particular shape. And so you can see it's a little bit wider here and made up of far more subunits here that hold it together in this stronger way. And here you can see they're stained green in this one. The nucleus is shown. And again, you can see that intermediate filaments are all over the place inside of the cell. Microtubules are the largest, and they form rigid internal skeleton for cells. So this is like the cytoskeleton part, but they can also act as a framework for motor proteins to move along. So when we see movement within the cell happening, that can actually be proteins that are walking almost along these microtubules. And there's some really cool animations and things that we'll see in class that demonstrate this, but this forms, it's almost like they're little elevator cars that are riding along the elevator cable, and the elevator cable would be these microtubules. So these are made up of alternating different monomer subunits, alpha and beta tubulin. So tubulin is the monomer of microtubules, which is kind of easy to remember. Again, not a term we're going to specifically quiz you on, but if you saw this in a question presented to you as a little story about a particular molecule that's a microtubule and it's got tubulin, whatever, we could write a question that includes the use of these things, and so it can be helpful to just sort of generally be aware of their existence, so we're not thrown off by questions about that. And again, we see another cell. They're stained blue in this case, and again, a network that is extending in many different directions within the cell. So they do show dynamic instability. So like the microfilaments, the microtubules grow and shrink on either end, and that's a process of polymerization. Polymerization is a term that comes up often in biology. Don't be thrown off by it. Polymerization, they are becoming polymers, right? So we are building up. Depolymerization is the removal of those monomers. So a lot of times the clues are just in the parts of the word to help us to understand. We hopefully by this point have a pretty good understanding of how monomers become polymers and how polymers can be broken down into monomers. That's what this is. That's what this is all about. Two major types of microtubules are used in cilia and flagella. So cilia being very short, but useful for movement, and then flagella are the longer, like big tails, and they have a snake-like movement that thrusts the cell around, and different microorganisms make use of that. And then there are different cells even in humans that make use of either of these as well, but not so much, not nearly the extent to what we see like in protists or bacteria. Here you can see some cilia on a cell, and these are the cilia again, but in a more traditional view. It's a transmission electron microscope view. Here's a scanning electron microscope view of the exterior of the cell. Just kind of a pretty cool picture given just the size of these things, the scale here, nanometer scale or micrometer. Okay. I'm not going to go into details about this 9+2 arrangement, doublets, and all of that. It's great biology. It's really cool stuff to gain a good understanding of. It's not in the scope of this course, but if you wanted to know more, there's a lot of really interesting things, and it doesn't really hurt to know more, just because the more specifics you learn, I think the stronger your ability to rely on the fact that there's pretty basic and expected chemical interactions going on all throughout the cell. And so you eventually, hopefully, in your science and biology career, you'll reach a point where the specifics will rarely phase you, just because even if we didn't go into like the specifics of how microtubules, for instance, are arranged, we should have some pretty good assumptions just from our basic understanding of chemically what needs to be going on, that we can digest this pretty easily. And here's just, it's a complicated structure. It's these hollow tubes with these little, what are called doublets here, circled all around. And there's these motor proteins which facilitate the movement along these things. And I think there's a picture here, yeah. So these little doublets here have connections between them. And then what happens is different types of motor proteins, in this case kinesin, or I'm sorry, no, I lied. Kinesin is the one that I'm going to show you a video of in class, where it's a representation basically showing this thing walking along microtubules and delivering a vesicle through the cell. It's really cool. Dynin and Nexin are the two that serve to facilitate this movement. So here we have Dynin, which is walking along one of these doublets. Now, if that's allowed to happen, then the doublets will move in opposite directions of each other. But in the case of cilia and flagella, there's this other protein called Nexin, which basically holds them rigid to each other. But then when the dynin starts to make them sort of pull against each other in opposite directions, instead of just sliding apart, they bend. And so it's a coordinated movement of that bending that causes the waving of the cilia and flagella. Here's the kinesin. Again, there's really cool computer-generated sort of models of how this works, and it's kind of creepy how much this thing really is just like legs walking and taking steps down this microtubule. So you can see some of these structures under the microscope, and the function can be observed within the cell, but they take a good opportunity in this section of the book to explore the idea that we have to be careful when analyzing structure and function, because we can observe things and we can make assumptions about what purpose they serve, but it still requires pretty extensive scientific investigation to really definitively make claims about whether that structure is in fact facilitating that function. And so there's a couple methods that they talk about, one being a method of inhibition, where you inhibit the production of a protein inside of a cell using various drugs or artificial or natural inhibitors to prevent the creation of that protein. And then if inhibiting it stops the function, then you have a pretty good support that protein was in fact, or that structure was in fact responsible for that function. Or maybe you inhibit the structure and the function still happens. Okay, good learning opportunity to say, oh, well, we made an assumption about this thing's function and we were wrong. Another type is mutation, so looking across different organisms that may have had genetic defects or things like that, and if the genes simply aren't there to even produce the protein or structure to begin with, if the function doesn't happen, then you could make a case for why that protein or structure was responsible. And the textbook ends this section with a little investigation, basically just demonstrating that, but I'll run through it just real quick. Again, none of the specifics of this really matter, but being able to do this kind of analysis is, an indicator of a good, hard-thinking AP Bio student. And so here there's an amoeba, which is treated, it's a eukaryote. It moves by extending its membrane using microfilaments, or at least that's our hypothesis, is that they're using the cytoskeleton to move. And there's a drug called cytoskeleton B, that is, it blocks microfilaments from being produced. And so they treat two amoebas, one with the cytoskeleton, and the other one receives an injection but doesn't get the drug. So that way the injection is not responsible for the effect. So you want to make sure everything's the same between these except for our one specific variable, which is the cytoskeleton. I can't say that. It's really hard. Cytoskeleton. So what they found was that the treated amoeba was alive, but it rounded up and it couldn't move anymore. So that, again, would support the hypothesis that this drug, which blocks the microfilaments, that the microfilaments, therefore, are responsible for movement in this amoeba. The control, meanwhile, continued to move. And here you can see they tested a whole bunch of different drugs: cytoskeleton, colchicine, cyclohexamide, and dinitrophenol. And reading through here, you can see what each of these does. The cyclohexamide inhibits protein synthesis, dinitrophenol inhibits ATP formation, and colchicine inhibits polymerization of microtubules. And so there's a lot that could be taken from something like this in all of this different data, because we can see that consistently, if the cytoskeleton B is present, whether it's in conjunction with other drugs or not, we see massive amounts of rounded cells. That is, cells that no longer are able to move. The cytoskeleton's not able to extend their little branching components to crawl along whatever environment they're in. And meanwhile, the other ones here, which are all key inhibitors of various components of the cytoskeleton, they don't have nearly the same effect. In fact, we see that no drug, there's still about 3% of cells that just round up and don't move, and that the colchicine and dinitrophenol do a little bit, but cyclohexamide didn't do anything. So that tells us a story of what's going on here, that even though colchicine inhibits polymerization of microtubules, which is a component of the cytoskeleton, clearly the amoeba cell movement is not related to microtubules, but it is related to microfilaments, as evidenced by this. So that's a lot of things to say about a pretty simple idea, but I think this is a pretty powerful example of how we use scientific data to learn new things about the cell, and that through repeated trials of treatments of different drugs and inhibitors, we can then piece together exactly what the function of these different things are. So that way, when we say that, like, oh, hey, there's microfilaments and there's 50 different kinds of microfilaments inside of the cell, you should be thinking, like, how the heck do we even know that? And how do we know what each of those does? And the answer is just so many experiments like this that have been done over primarily, I guess, the past 100 years, but in some cases even longer than that. People have been trying things out and learning things slowly but surely, and certainly in more recent time we've exploded with the ability of technology and ubiquitous availability of information and all of that, and today we understand so much more. So, cytoskeleton is a very important component of the cell. It's important for us to be aware of it and aware of, like, the big level functions of it, but no specific real vocab from this section in terms of, like, specific types of proteins. You're not going to have to differentiate between microtubules, microfilaments, intermediate filaments in any way in any questions that we ask. But like I said, you'll see these terms sort of adjacent to the main point of what we are talking about. And so don't want you to be thrown for a loop by just words popping up. But so that's enough on this section. The next section is extracellular components. Let me know if you have questions.